Thank you and welcome to Synthego's webinar, Troubleshooting ICE, Suboptimal Error Messages. My name is Mike and I am a technical support scientist at Synthego. In this webinar, we will discuss common issues and complications when working with ICE resulting in suboptimal ICE messages and how to troubleshoot them. Here is what we will cover. First, we will take a look at the limitations of ICE. Next, we will take a look at what suboptimal error messages look like in ICE. Lastly, we will discuss possible causes and suggest solutions to fix the suboptimal error messages ICE is producing with your results. Remember that you can download a PDF copy of the full slide deck to follow along and take notes during this webinar adventure. Let's get started. ICE can analyze a variety of SP-Cas9 mediated edits such as knockouts, small knock-ins, SNVs, or codon swaps. However, it does have some limitations. When you are trying to knock out a gene using a single guide RNA, the largest deletion ICE can detect is up to 40 base pairs. When using multiple guides for your knockout, ICE can analyze deletions of up to 150 base pairs. It should also be noted that 3 guides are the most that can be input into ICE for your analysis. For knock-in experiments, ICE has the capability to detect insertions up to 270 base pairs. ICE also assumes you are using SP-Cas9 nuclease and will place the cut site 3 bases upstream from the 3' end of the guide. Lastly, Sanger sequencing always has some inherent noise. Because of this, ICE cannot clearly determine if sequences present at 5% or less are background noise or if they are truly present. Even if these sequences are real, the sensitivity of Sanger sequencing is very low in this range. We therefore recommend cautiously evaluating any sequences that are present at 5% or less. If you see a status of succeeded in orange with an error message below as seen here, this is considered a suboptimal ICE analysis. The different error messages will indicate what needs to be improved to have ICE successfully analyze your sequence files. 2 common examples of these suboptimal error messages and the possible causes why the messages occurred will be covered on this slide. Each of these error messages have recommendations corresponding to them Synthego recommends applying to troubleshoot the analysis. The first one reads, padding alignment window to be at least 40 base pairs in length from the start of the sequence. This is caused by the primers being too close to the cut site and not allowing the Sanger sequence reaction to stabilize. This can be fixed by redesigning your primers and adjusting the space between primers and cut site to be at least 150 base pairs away from the guide sequence. Another common suboptimal error message is no PAM up or downstream of the guide sequence. There are 2 possible causes for not having a PAM sequence being recognized. One possible cause is having an incorrect guide sequence being entered into ICE initially. Similar to other messages with incorrect guides being entered, making sure you input the correct guide sequence into ICE is essential. This sequence should be the same exact guide sequence you introduced into your samples to create the genome edit. The second possible cause is that Cas nuclease enzyme you used was not SP-Cas9. ICE assumes you are using SP-Cas9 nuclease and will place the cut site 3 bases upstream from the 3' end of the guide. So if you use other Cas nucleases and ICE still proceeds through with analysis, we cannot guarantee the accuracy of the ICE results. If you would like additional resources of ICE, how to analyze results, and other webinars, feel free to glance over these links to help you further. All of our resources are freely available on our website and are great tools to help you get started with your experiments. Here at Synthego, we would like to help you with your CRISPR needs anytime. Please feel free to reach out to us and we will be happy to assist you. I would like to thank you for your time, and please visit the Help Center website at www.synthego.com for more information.